Does it always seem like you have dry mouth? Does it always seem like you have no energy? Does your urine taste like sugar? If you answer yes to those questions, then you might be a type 1 diabetic. But don't worry, we have just the product for you.
In a normal functioning body, insulin is secreted by beta cells of the pancreas. It is a peptide hormone made up of a sequence of 51 amino acids. Generally, insulin allows the body to use sugar from the blood for energy and to store it for later uses. Its purpose is to lower blood glucose levels by promoting cellular uptake of glucose, primarily in muscle and fat, and to promote storage of energy through metabolic pathways, such as glycogen synthesis in the liver. In type 1 diabetes, the pancreas does not function correctly and cannot synthesize insulin. This happens because the immune system incorrectly targets and destroys the beta cells which produce insulin. Therefore, repeated injections of insulin can be an effective remedy for people with type 1 diabetes. Prior to being produced synthetically, mass quantities of insulin were derived from pancreatic cells of animals, such as pigs and cows. This production, however, had several drawbacks. While very similar to human insulin, the hormone from animals is slightly different. Pig insulin differs from the human's by only 1 amino acid, while cow insulin differs by 3. In some cases, this can lead to rejection of the insulin by the human immune system. The animal insulin also lasted for a shorter period of time. Additionally, around 2 tons of tissues from pigs were needed to yield just 8 ounces of insulin. Potential issues could be encountered with vegans, for example, people who prefer not to use animal products. Therefore, a new source of insulin was warranted. Luckily, scientists have discovered a way to utilize bacteria to produce bioequivalent synthetic insulin. There's a lot to the process of producing synthetic human insulin, and we're going to give you our inside scoop on how it is done. The bacterium that is used in this process is one of our well-known favorites, Escherichia coli, or more commonly known as E. coli. The process begins by growing massive quantities of E. coli in fermenters and applying the optimal growth conditions that allow that bacterium to grow at its fastest rate.
An important side note, growth conditions are important in microbiology. Things such as temperature, pH, salinity, availability of nutrients, and many other factors contribute to how microorganisms grow, where they're going to grow, and how they're going to behave.
In order to ensure that only the insulin-producing E. coli are grown, a special plasmid is designed in order to include an antibiotic resistance. One example of these antibiotics is tetracycline. It is then introduced into the vat to kill any extraneous microorganisms.
Take a walk with me. There are 2 important proteins in the next step of our process. According to microbiology and evolving science, a repressor protein is a protein that binds to the regulator sequences to prevent transcription of the target gene. And the second is an inducer protein, which is a chemical that binds to the repressor protein, allowing that target gene to be expressed.
How this is involved in the insulin process is that E. coli has not been making insulin during the other steps of this process due to a repressor protein. So, an inducer is then added to the bacterium that allows them to make the proinsulin protein. The insulin that is produced is not released into the vat. Instead, the insulin is stored inside the cell in an insoluble inclusion body, where chaperone proteins fix any mistakes to the protein that could have occurred during translation. After that, the cells are centrifuged at around 6,700 Gs, and protein pellets are formed from the former inclusion bodies. The insulin is then purified once again through the use of liquid chromatography columns and is then combined with zinc in order to form crystals, which are then reconstituted later into insulin's final liquid form.
And there you have it. That's the process of how insulin is made. Recombinant DNA has allowed processes like these to become possible, and just goes to show how microbiology has and will continue to have a positive impact on the world that we live in today.